welcome to a new video class. In this video lecture, I like to explain the process that is link reaction. We know that when we take the case of cellular aerobic respiration, you can see the breakdown of one molecule of glucose into two molecules of pyruvic acid. So that is a partial breakdown of glucose into two molecules of pyruvic acid. During that process, we will get a net energy of 7 ATP. That is 4 ATP and 2 reduced NAD, that is NADH2. Whereas this glycolysis takes place within the cytoplasm of eukaryotic cell. So next, this pyruvic acid or pyruvate then moves into the mitochondrial matrix through a special type of mitochondrial pyruvate carrier or mitochondrial pyruvate transporter. So with the help of this mitochondrial pyruvate carrier or mitochondrial pyruvate transporter, the pyruvic acid produced within the cytoplasm moves into the mitochondrial matrix. From there, that pyruvate or pyruvic acid converted into, we know that two pyruvic acid created within the cytoplasm during the breakdown or during the glycolysis of one glucose molecule. So that two molecules of pyruvic acid then converted into two molecules of acetyl coenzyme A. It is a 2-carbon compound, and like that pyruvic acid is a 3-carbon compound. So the conversion of pyruvic acid into acetyl coenzyme A with the help of pyruvate dehydrogenase complex, with the help of an enzyme called pyruvate dehydrogenase complex. So with the help of pyruvate dehydrogenase complex, this pyruvic acid converted into acetyl coenzyme A. During that process, we will get two molecules of carbon dioxide and two molecules of reduced NAD, that is NADH plus H plus. So this is the broad concept of link reaction. Actually, the term link reaction given to this process due to a specific reason. That is, we know that these pyruvic acids are formed as a result of glycolysis in the cytoplasm of cell. So that glycolytic product called the pyruvic acid cannot directly enter into the succeeding or cannot able to enter into the next stage of aerobic respiration, that is Krebs cycle. So in order to enter this pyruvic acid into the Krebs cycle, which is taking place, which is going to take place within the mitochondrial matrix, we need a special kind of reaction. That reaction is called link reaction. Hence, we use the name link reaction. That is a reaction which connects the glycolytic product, that is pyruvic acid, into the Krebs cycle. That is the major step of aerobic respiration. So due to that reason, we call this process as link reaction. That is about the link reaction. Next, we need to study the details of this pyruvate dehydrogenase. Actually, pyruvate dehydrogenase is a enzyme complex consisting of 5 cofactor and 3 apoenzymes. So it is not a single enzyme. Actually, it is an enzyme complex. So if we try to study all the details regarding this enzyme complex, then we will get the whole idea about the conversion of this pyruvic acid into acetyl coenzyme. Next, I am going to explain that detailed topic regarding with the enzyme complex, that is pyruvate dehydrogenase complex. So this is the main component of pyruvate dehydrogenase complex. We can say that PDH or PDC, PDC or PDH. This pyruvate dehydrogenase complex mainly consisting of 5 coenzyme cofactors and 3 apoenzymes. These are the protein part and these are the non-protein parts. So these 5 coenzyme consisting of TPP, that is thymine pyrophosphate, that is called TPP. Actually, it is a derivative of vitamin B complex, that is vitamin B1. It is a derivative of vitamin B1. And second one is called coenzyme A. Actually, that coenzyme A is a 21 carbon compound. And like that, third one is called flavin adenine dinucleotide, that is FAD. And fourth one is called nicotinamide adenine dinucleotide, that is NAD, nicotinamide adenine dinucleotide. And fourth one is called lipamide. Actually, lipamide is a 8 carbon compound. You can see the structure of molecular formula of that. It's not an accurate structure, it's just a molecular formula of lipamide. You can see 1, 2, 3, 7, 8 carbon, and like that. You can see 15 hydrogen, 1 nitrogen here, and 1 oxygen here, and 2 sulfur here. So actually, this lipamide is existing in oxidized form. Suppose if it exists in a reduced form, then 1 hydrogen atom will come here and 1 hydrogen atom will come here. So this won't. I remove this one. So now this is in the reduced form of lipamide. If you remove these 2 hydrogen and give a single covalent bond here, now it becomes an oxidized form of lipamide. So this is about the 5 enzymes of pyruvate dehydrogenase complex. When we move into the protein part, that is 3 main apoenzymes are present for pyruvate dehydrogenase complex. That is, one is called pyruvate dehydrogenase, and second one is called dihydrolipoyl transferase, and third one is called dihydrolipoyl dehydrogenase. So due to the presence of these components, we can call it as pyruvate dehydrogenase complex. These complex catalyze or convert 2 molecules of pyruvic acid into 2 molecules of acetyl coenzyme by releasing 2 molecules of carbon dioxide and by generating or reducing 2 molecules of NAD. So this is about these enzymes. Now we need to study how these enzymes complex convert 2 molecules of pyruvic acid into 2 molecules of acetyl coenzyme. So it is better to draw a diagram or a pathway in order to understand the mechanism of converting pyruvic acid into acetyl coenzyme within the mitochondrial matrix. So here I am going to draw the structure or the mechanism of conversion. So you just wait here, I will explain all those things very clearly. Here you can see the representation of acetyl coenzyme formation from pyruvate or pyruvic acid. That reaction is called Linde reaction, and that reaction takes place with the help of 3 apoenzymes, that is pyruvate dehydrogenase here, dihydrolipoyl transacetylase here, and dihydrolipoyl dehydrogenase you can see here. and also 5 coenzyme, that is thymine pyrophosphate, like that. You can see here this lipamide, and third one you can see FAD, and fourth one you can see NAD, and fifth one you can see coenzyme A. This is coenzyme. I'll write. This is coenzyme A. This is CoA. This is coenzyme A. So one is here, and one is here, and like that one is here, and like that one is here, and like that one more thing. One is here. This is the 5 coenzyme, and this is the 3 coenzyme. These components actually participate for converting this pyruvic acid. This is called pyruvic acid, glycolytic product pyruvic acid, into acetyl coenzyme A. You can see this is called acetyl coenzyme A. So we are going to study how this pyruvic acid converted into acetyl coenzyme A with the help of these 8 factors, 3 coenzyme and 5 coenzymes. So first of all, this is one molecule of pyruvic acid. We know that glycolysis of one molecule of glucose gives 2 pyruvic acid. Here I only represent one molecule of pyruvic acid. First of all, one molecule of pyruvic acid undergoes oxidative decarboxylation. Here one molecule of pyruvic acid undergoes oxidative decarboxylation, means removal of one carbon dioxide. This is CO2. One decarboxylation in presence of an enzyme. That enzyme is called pyruvate dehydrogenase, and we get, so decarboxylation of pyruvic acid will use one molecule of carbon dioxide, and at the same time this thymine pyrophosphate, which is a derivative of vitamin B1. So this thymine in presence of thymine pyrophosphate, decarboxylation occurs in presence of thymine pyrophosphate and enzyme 1. We get a new product that is called hydroxyethyl thymine pyrophosphate. This is called hydroxyethyl thymine pyrophosphate. So that is the role of enzyme 1. So after the formation of hydroxyethyl thymine pyrophosphate, next the enzyme 2 catalyzes the second reaction. During that process, mainly oxidation takes place. These 2 hydrogen, this one H and this one H will remove. So oxidation of this hydroxyethyl thymine pyrophosphate in presence of enzyme 2 gives another compound that is called acetyl lipamide. You can see CH3CO, acetyl lipamide. So here what happened is this thymine pyrophosphate get replaced by this lipamide. This is called lipamide in oxidized form. Here you can't see the hydrogen atom here. That is why it is an oxidized state. So here thymine pyrophosphate get replaced by this lipamide, so that you can see this thymine pyrophosphate here, and this lipamide connected here, so that we get CH3CO, SLSH. So oxidation occurs during the in presence of enzyme 2. During next stage, you can see this lipamide react with acetyl coenzyme. This lipamide react with acetyl coenzyme, and we get coenzyme. This lipamide, this acetyl lipamide react with coenzyme. Then we will get acetyl coenzyme. During that process, this lipamide from the acetyl lipamide releases in a reduced form. So here the lipamide converted into a fully reduced form. You can see hydrogen atom along with the sulfur. So they are fully in reduced form. And that reduced form of lipamide then again converted into oxidized form of lipamide with the help of third enzyme, that is called enzyme 3. That enzyme 3 is a dehydrogenase enzyme. You can see dihydrolipoyl dehydrogenase. So from the word itself you can see it catalyzes dehydrogenation of dihydrolipamide. In this lipamide you can see 2 H hydrogen atom, that is called dihydrolipamide. So it removes 2 hydrogen atom from this reduced form of lipamide, and that 2 hydrogen atom or H2 is used for reducing this FAD into FADH2. But this FADH2 then again oxidizes into an FAD by releasing 2 hydrogen atom. That hydrogen atom is used for the reduction of the final product, that is NAD into NADH2. That NADH2 is the main energy of mitochondria or main substrate of mitochondria for conducting electron transport system, thereby that mitochondria can able to produce 2.5 ATP from this one NADH2. Like that, our glycolytic product gives 2 pyruvic acid from 1 glucose. Here I only represent 1 pyruvic acid. If it is 2 pyruvic acid, then we will get 2 NADH2. So from 2 pyruvic acid, we will give 2 NADH2 during the process of Link reaction, and also we will get 2 molecules of carbon dioxide also. So the main products of this Link reaction is the release of 2 carbon dioxide molecule and the formation of 2 NADH2. And like that we will get thymine, the thymine pyrophosphate will release. Like that lipamide will also release. So these are all the things related with the Link reaction which is taking place inside the mitochondrial matrix. Actually the source, that is pyruvic acid, produces it in the cytosol, then that pyruvic acid move into the mitochondrial matrix with the help of a special type of carrier protein found on the inner membrane of mitochondria. That carrier protein is called mitochondrial pyruvate transporter or mitochondrial pyruvate carrier. So this is about the Link reaction. So the total energy gained during this Link reaction is equal to 5 ATP, that is 2 NADH2. Next, we are moving into the regulation of Link reaction. You know that what is Link reaction? That is 1 pyruvate or 2 molecules of pyruvate is converted into 2 molecules of acetyl coenzyme. 2 molecules of acetyl coenzyme. This reaction is characterized by an enzyme pyruvate dehydrogenase complex. Here I wrote PDC to understand where is the pyruvate dehydrogenase complex. That complex contains enzyme 1, enzyme 2, enzyme 3. Actually, this pyruvate dehydrogenase complex not contain single copy of E1, E2, E3. Instead, it contains approximately 20 to 30 copy of E1, 60 copy of E2, and 6 to 8 copy of E3. This Link reaction regulated mainly by covalent modification. One is covalent modification and second one is called allosteric mechanism, mainly substrate level inhibition. That is, acetyl coenzyme can be able to inhibit this enzyme. That is called substrate level inhibition or feedback inhibition. So covalent modification and substrate level inhibition are the main regulatory mechanism in case of Link reaction. Here we need to study 2 enzymes. One is called pyruvate dehydrogenase. That is, pyruvate enzyme 1 is called pyruvate dehydrogenase. That pyruvate dehydrogenase phosphatase and pyruvate dehydrogenase kinase. These are the 2 main enzymes which regulate the Link reaction, especially regulate the first enzyme of Link reaction. If these enzymes regulate the first enzyme of Link reaction, then we can say that this enzyme can able to regulate the whole process. Here, this phosphatase enzyme dephosphorylate the enzyme 1. If it dephosphorylate the enzyme 1, the dephosphorylated enzyme 1 become highly active. Then that active enzyme can able to catalyze this reaction very smoothly. Suppose instead of phosphatase, if PDH kinase phosphorylate this enzyme 1, then the phosphorylated enzyme become inactive. The inactivated E1 cannot able to catalyze this reaction. So dephosphorylation of enzyme 1 results the smooth working, and phosphorylation of enzyme 1 results the inhibition of this process. And one more thing about this regulation is that NADH2, ATP and acetyl coenzyme can able to, these three things can able to stimulate PDH kinase. So if it stimulates PDH kinase, then the stimulated PDH kinase or activated PDH kinase phosphorylate is enzyme 1. Then that phosphorylated enzyme 1 results the inactivation of E1. If E1 get inactivated, the process will stop. Because here the main logic behind here is that presence of NADH2, ATP and acetyl coenzyme in a cell indicate that that cell contains sufficient amount of energy, and also the cell can able to produce more amount of energy by using acetyl coenzyme also. So the cell need not produces further energy because the cell already contain more amount of energy. So at that time cell need to stop this process. So that is why it activate and phosphorylate, thereby phosphorylate enzyme 1 and that inactivated the process. Suppose if the cell contain magnesium 2 plus, calcium 2 plus and AMP, presence of these factors in a cell indicate that the cell needs to produce energy. The cell contain limited amount of energy. The cell need to produce energy. So in that condition, magnesium 2 plus, calcium 2 plus, AMP stimulate or activate phosphatase. That activated phosphatase then dephosphorylate enzyme 1. Dephosphorylated enzyme 1 is highly active. It catalyzes the reaction very smoothly. So this is about the regulation of link reaction. And one more thing about this link reaction is that PDC, pyruvate dehydrogenase complex, is a crucial enzyme which is an irreversible. We cannot able to produce acetyl coenzyme. We cannot able to produce pyruvic acid from acetyl coenzyme. That is, it is an irreversible reaction. So if pyruvate is converted into acetyl coenzyme, then the fate of pyruvate determined. This acetyl coenzyme can move into Krebs cycle, or it can move into the fatty acid synthesis, or it can move into the gluconeogenesis also. Okay, this is about the regulation of link reaction. Thank you.